Hello, ladies and gentlemen. Today I'm going to be talking about Chapter 12 in our textbook. We are getting towards the end, if you believe it or not. The next 5 chapters, 12 through 16, all cover the neurological system. Today I'm going to be talking, as the author calls it, nervous tissue. I don't particularly like that name. I would rather call it tissue of the neurological system. But he's the author and he gets to call the shots here. Now, hopefully you have already printed off the figures, the tables that I gave you a list previously, because I want you to, again, to be able to look at these figures while I am talking about them and trying to hit the high points. The first thing on the list on page 471, of course, is the study guide. This is a summary of the most important parts of this chapter that we want you to learn about. So, with that in hand, we're going to first look at section 12.1, which is called an overview of the nervous system. Now, there are 2 basic different organ systems in our body that have the jobs of communicating with all other systems of the body. These are the endocrine system, which we'll cover in the next course in Biology 251, and the nervous system or neurological system that we will start covering today. The nervous system has 2 major subdivisions. The central nervous system, which is comprised of the brain and the spinal cord, and the peripheral nervous system that consists of everything else. All of the nerves and all of the nerve ganglia that are outside of the central nervous system. And then if you look at figure 12.1 and the discussion there on page 437, you will see how this peripheral nervous system is then functionally divided down into other subheadings. Now, we're going to talk about section 12.2. What is a neuron? N-E-U-R-O-N. A neuron is the name given to any cell of the nervous system. Neuron. All of these cells have 3 fundamental properties that are very important. One is excitability. That means that they are able to respond to things that occur outside of themselves. They respond to environmental changes. Only muscle tissue and nervous tissue can do this. The second is conductivity. When a neuron responds to a stimulus, this stimulus, or the electrical system, is quickly transmitted and conducted to other cells at different locations. And then finally, secretion. Now, when the signal reaches the end of the nerve fiber in the terminal axon, it secretes a chemical called a neurotransmitter that crosses over a gap and stimulates the next cell in the line. There are 3 general classes of neurons. One are the sensory neurons. These are specialized to detect multiple different stimuli, such as light, heat, pressure, chemicals, and they transmit this information to the central nervous system. These are called afferent neurons, afferent. The A we can think of as standing for at. They go towards or at the central nervous system. Then there are many, many different types of interneurons. These lie totally within the central nervous system. They receive signals from other neurons and then carry out what's called their integrative function. They can process, store, and retrieve information and make decisions about how to use it. About 90%, your author says, of all the neurons in the body are interneurons. And then finally, there are efferent, EFF, efferent neurons. These neurons send signals away from the central nervous system, predominantly to muscle and gland cells. These are the effector cells. Now, I want you to look very, very closely at figure 12.4 on page 439. This is a typical or a representative neuron. At the top, you will see a structure that has many, many tiny branches off of it. In the very center of that structure is the nucleus, and the dark staining material in the middle is the nucleolus. Surrounding it is the cell soma, is called S-O-M-A. This is where all of the other things take place in any typical cell. The little branches that branch out from the soma are called dendrites. These receive impulses from other locations. The soma and the nucleus decides what to do with these impulses. And then at the bottom is called a trigger zone. This is where the final decision is made whether or not to send a nerve impulse through the axon. The axon is a long branching part of the cell that leads away from the cell body. In one place you see an axon collateral. That is a side branch. But with the big red arrow, you can see the direction of the signal transmission. And it goes all the way down to another set of branches called the terminal arborization. Arbor as in trees. This is a representative neuron. Remember, the dendrites gather information. They send it to the cell body and the nucleus of the neuron. And then, if it's appropriate, impulses are sent on down the axon to the terminal arborization. And we'll look at what happens there in just a moment. You can see an excellent photomicrograph in part B that shows that we're not making these drawings up. That's what an actual neuron looks like in the microscope. Now, at the distal end, each little branch of this terminal arborization has what's called a synaptic knob. This is a small swelling that forms a junction with the next cell, and it contains synaptic vesicles. If you will recall that we looked at one of these when we talked about how muscle cells are stimulated. It is the same thing here. These synaptic vesicles are full of different types of chemicals called neurotransmitters. Now, the terminal axon looks a little strange here because it is a myelinated fiber. The myelin, again, we will talk about in just a moment. It allows the nerve signal, the nerve impulse, to be transmitted much, much faster than in an unmyelinated fiber. And for your information, whoops, cattail, you don't need to see. The myelination actually makes the tissue look white. If you've ever seen parts of a brain, you know that there are white cells and gray cells. The white cells look white because of the myelination. In section 12.3, we will discuss the other different multitudes of supportive cells called neuroglia. There are 6 different types of these, and your author estimates that the neuroglia make up about 50% of the volume of the nervous tissue. There are an estimated 1 trillion, 10 to the 12th neurons in the nervous system, and about 90% are neuroglia, or sometimes called glial cells. There are 6 different types of glial cells. These are listed very succinctly in table 12.1. and they are described on pages 442 and 443. Figure 12.6 at the top of page 443 shows some representative different types of these glial cells. Each looks different, and each has a very specialized function. Let me just tell you how to pronounce these and something very important about each one. Oligodendrocytes, number 1. These help form the myelin sheath that wraps around the axons, oligodendrocytes. Second type are ependymal cells. These line the cavities of the brain and the spinal cord, and they produce cerebrospinal fluid. Thirdly, or number 3, are microglial cells. These are small macrophages that actually develop from monocytes, which is a specific type of white blood cells. They wander around freely, getting rid of dead and damaged tissue. Astrocytes are the most abundant of the glial cells, and they have numerous different functions, as you'll see listed there. Finally, there are Schwann cells and satellite cells. The Schwann cells are actually the cells that wrap around the nerve fiber, and they also produce a myelin sheath that we will talk about here just in a minute. In fact, page 444 talks about myelin. Here's how it is defined. Myelin sheath is a spiral layer of insulation around a nerve fiber formed by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. The production of this sheath is called myelination, and it basically helps the peripheral nerves and essential neurons to be able to transmit impulses much faster. In between these different pieces of wrapping, you will see small gaps called the nodes of Ranvier. That's a French word. Wish I spoke French. They're also called internodes. And these have a very significant function in conducting impulses down the nerve fibers. On page 446, 447, you'll see a little discussion of regeneration of nerve fibers. Now, you might have heard that many different parts of the nervous system cannot regenerate. That's why if there is a serious damage to the spinal cord, for example, or an area inside the brain, it cannot regenerate. However, sometimes a peripheral nerve fiber can regenerate if its soma is intact and at least some of the neurolemma remains. And if you're interested, you will see how this happens in a 6-step process there on page 447, 446, 447. Skipping on now to section 12.4, the electrophysiology of neurons. Neurons only communicate with other cells by virtue of electricity, if you will. Electrical potentials and nerve impulses are what allow the transmission of important information. By definition, an electrical potential is defined as the difference in the concentration of charged particles between one point and another. This produces a certain amount of potential energy. Under the right circumstances, that is when the nerve impulse occurs, then an electrical current is sent on down the branches of the neuron. Looking now at figure 12.11, page 449. The primary electrolytes that are responsible for the resting membrane potential in a neuron are sodium and potassium. You can see that, as anywhere else, inside the cell there is a huge number of potassium ions and a few sodium ions. In the extracellular space, it's just the opposite. There are many, many more sodium neurons and then potassium neurons, or not neurons, sodium or potassium ions. Now, there are also many different larger anions that are present inside the cell. And all of this makes up what is called the resting membrane potential. And this is about a minus 70 millivolts in a neuron. The difference in charge between the inside of the cell and the outside. This negative value of a negative 70 millivolts, when this neuron is at rest, is because there are more negatively charged particles in the intracellular space than there are outside the space. Now, when this electrical potential changes due to opening and closing of sodium channels or potassium channels, then there is the very distinct possibility of a nerve impulse resulting. You will read there about how this potential can change, how the sodium-potassium pump functions in this phenomenon, and the importance of it. Figure 12.12 shows the excitation of a neuron by some chemical stimulus, and this changes the resting or the membrane potential. There are local potentials that do not go anywhere. They stay right there at the cell level. And there are action potentials. These are the ones that, when a dramatic change is produced, these can be transmitted through other parts of the cell to other cells in the body, other neurons or muscle cells, etc. Please look now at figure 12.13, page 451. You will see the diagram of an action potential. This is very different from just a local potential. I'm not going to go through all of the 7 details there, but if you're going on into, or not 7 details, but the 7 steps that cause this rapid upswing in the potential and then down falling of this potential from -70 to +35 millivolts, and then down to even less than -70. This is an action potential, and it is caused by various different sodium and potassium channels opening and closing at certain times that cause this rapid depolarization, becoming more positive, and repolarization when it becomes more negative. If you are going on into any sort of a medical or scientific field, it would behoove you to look at this in more detail and try to remember what occurs at each of these stages of the action potential. Now, if you look at figure or part B, sketch B of figure 12.13, you will see something interesting also. When the neuron repolarizes, it actually goes more negative than the -70 millivolts. This is called hyperpolarization. We'll talk about that in just a moment. 3 very, very important things about an action potential that you should know. And these are in dark black letters on page 451. All action potentials follow what's called an all or none law. When the membrane potential depolarizes past what's called the threshold, look back at figure A in figure 12.13, you'll see a blue dotted line at -55. That is the threshold of this cell. If the depolarization occurs and the voltage differential is less than -55, more positive, then this all or none law takes place. The stimulus depolarizes the neuron all the way to about plus 35, and the entire part of the neuron is stimulated. The action potential that is generated is also called non-decremental. That means that it travels away from the cell body, on down the axon, all the way to the end of the nerve fiber with this same positive 35 millivolts. It does not become any less positive or more negative as it is transmitted. And these action potentials are also irreversible. If a neuron reaches threshold, the action potential then goes all the way to completion, and this nerve impulse is transmitted. It does the size and shape of the depolarization, repolarization do not change. Figure 12.14 is very interesting, shows sodium and potassium channels as they open and close during an action potential. I want to talk very briefly and define what are called an absolute refractory period and a relative refractory period. If you look at figure 12.15 in the orange, light red, whatever color it is, this is the absolute refractory period. That means that during that time period, very, very short time period, this particular neuron absolutely cannot be stimulated again, no matter how strong is the stimulus. When the repolarization goes below negative 70, then there is a short amount of time that is called the relative refractory period. During this time period, this particular neuron can be stimulated again, but the impulse needed in order to do that has to be a larger impulse than originally caused this action potential to form. Now, very briefly, difference between myelinated and unmyelinated fibers. All I really want you to know is that myelinated fibers are able to transmit nerve impulses much, much faster than unmyelinated fibers. And this phenomenon is called saltatory conduction. And that is explained in very great detail in figure 12.17. It has to do with the myelination and these nodes, remember the nodes of Ranvier. This allows this nerve impulse to be transmitted very, very quickly. Starting now into section 12.5, we talk about synapses. Skip just for a moment over to figure 12.20 on page 457. So here you will see the sketch of a synaptic knob. This is a knob at the end of each axon. You'll see that between the synaptic knob and the postsynaptic cell, whether it's a muscle cell or here it's another neuron, there is a small space called the synaptic cleft. In order for this nerve impulse, the depolarization, to jump across that cleft, there have to be neurotransmitters. And these are chemicals that are stored in many, many different vesicles inside of this synaptic knob. In this particular figure, 12.20, they are shown as sort of a light grayish color. And you will see them joining on to the outer cell membrane in the synaptic knob, or at the edge of the synaptic knob, and then releasing their neurotransmitters, which go across the space and join into very specific receptors in this postsynaptic cell. Now, when we talked about muscle fibers being stimulated, we talked only about one specific type of neurotransmitter, if you'll recall, and that was acetylcholine. It is a chemical molecule formed from acetic acid and the molecule choline. It has one very specific function, and that is to stimulate muscle. There are also different amino acid neurotransmitters and monoamine biogenic amines. Look, if you will, at table 12.3 on page 458. And there you will see the major types of neurotransmitter. And oops, I forgot something here. I said that there were 3 major types. I lied. There are actually 4. The 4th group are called neuropeptides. These are chains of from 2 up to 40 different amino acids. Figure 12.3 shows the different neurotransmitters, the names of them. It shows where they function. It also shows some of the typical chemical structure. Again, you don't have to know all of this, but you may see one question on the unit exam. I don't know. In figure 12.22, you'll see an identical picture, or almost identical to what you saw in muscle tissue. Here you will see the transmission at a cholinergic synapse. This is a synapse that uses only the neurotransmitter acetylcholine. And it is abbreviated capital A, capital A, capital C, small h, acetylcholine. And there are 5 steps in how this functions, how this occurs. They are listed in detail on the bottom of page 459. Now, going on very briefly to what is section 12.6, neural integration. Up to this point, we have only talked about nerve transmission in one particular neuron, from the dendrites to the soma on down to all of the synaptic knobs. But as we well know, there are millions of different neurons, and this allows for the nervous system, primarily the central nervous system, to be able to do numerous extremely important different things. And I'm going to leave that up to you to cover briefly, to read about summation of signal, facilitation, inhibition, neural coding. Something more interesting about neural coding. For example, how does the central nervous system know if a pain that's caused in a finger is just a mild amount of pain, or a moderate amount, or a very severe amount of pain? This is done because of neural coding. If it is a very severe pain, these nerve impulses will occur much, much, much faster than if it is only a mild pain. You will also see at the very end some short discussions about memory. How are memories formed? And I'm going to leave that up to you to read and think about. And if you have questions, of course, to contact me. I am so-called, quote, expert, unquote. Okay, that is all of nervous tissue. Remember the tissue part. We will start then into chapter 13 in a little bit, talking about the spinal cord, the spinal nerves, and some different what are called somatic reflexes. The spinal cord is part of the central nervous system. The spinal nerves are part of the peripheral nervous system. Thank you for your attention, and again, I hope that this is helping you somewhat.